Ever wonder what makes the greats great? What makes the successful successful? What makes the brilliant brilliant? Our Tuesday meetups with the celebrities of pharma industry and science are your one-stop shop to all these answers and more. Join us for Pies of Life, an initiative of the Biopatrika Industry Mentorship Program, where we bring your dream mentors to you.
All right, so good evening, good morning, everyone. Today is a special day because we are turning ritual into tradition, and today marks the first episode of Narain Talks by Biopatrika. This Tuesday, we've prepared to be immersed into the world of biologics. We have the Associate Vice President for Product Development at Biocon Biologics as our guest today. His portfolio includes the development and lifecycle management of insulin analogs and monoclonal antibodies. An accomplished scientist, hardworking visionary, and desh premi, he is one of the few who moved back to India after his PhD at the University of Buffalo. You know of him, but now you will know him. Welcome to a Te with Karthik Ramani. Karthik, I'd like to invite you to tell us your life story in your words now.
Sure. So first of all, thank you everybody that you're going to spend some time here for the next about an hour, 45 minutes to an hour. And thank you, Naren, as always. Who's always gracious and always an enabler in terms of trying to understand what goes on. So where do I start? So maybe, actually so let me start. I was born in Chennai. I was brought up in Chennai. My schooling was in Chennai. So, pretty much I never left Chennai until I went to Pilani for my undergraduate studies. So pretty much just semi-joined family. Usually people used to visit us because most of the family members were in and around Chennai. And so I'd gone in and around Tamil Nadu, but never went out until Pilani. And I pursued my B pharmacy and my master's in biological sciences. Actually, I wanted to become a medical doctor, but somehow I didn't clear a single exam that happened during that year. So it was not very fun at that time. And essentially then I became so used to basically not getting through any of the exams. It was like, okay, fine, let me just go and write whatever is possible and let me see whether it was all Hail Mary and hopefully one of them will return the stick, but nothing happened. So, and then what happened, I think that was also the time the Mandal Commission had kicked in 1993. I think that was our batch which got impacted by Mandal Commission. Our first batch, that is when VP Singh government implemented the Mandal Commission, the reservation policy all across India. And many of the overzealous government, including the Tamil Nadu government then, actually ended up making it as 69% of reservation and 31% of whatever they call it, the so-called other caste. And incidentally, the Tamil Nadu also kind of did a unique thing. What they did is, you always have a good mix of people, even the so-called, I don't believe in caste, but even in the so-called backward or most backward caste, you really have very bright people who actually do wonderful things. And so when they compete and automatically when they do well, they come to the general category, that is our category. So that more people can basically come into the reservation category. So like this, what happened is essentially we became 1% or 2%. So, and eventually I did not, even the state exams, whatever cutoff was there, I was not meeting those expectations. So I decided to, actually take I did BSc chemistry in Vivekananda College. For about 6 months in Chennai. And incidentally, that was the only college which was a totally Brahmin college, and that was the only college which gave me seat also. The rest of the college refused to give seat also. So there was another, I would say, unexpected turn of events when I graduated in 92, 93. Then I reapplied for BITS, and BITS was the only institute in India where they allowed us to, there used to be a small pool of applicants who can apply for the second semester. And so somehow I managed to get in. And so I was there for about 5.5 years. Fabulous time, absolutely fabulous time. I didn't know what I wanted to do in life, pretty much. And I was also proudly one of the only student in the campus who did not get a campus interview job. I didn't attend any interview. So I refused to attend the interview, and I think 2 or 3 days before or rather about the last week of our exams, the admissions committee officer called me, and he said that, do you know why I have called you? I said, I don't know. He said, you're the only student in the entire batch who have not attended a single interview. So why don't you attend? Why don't you do something? What do you want to do in life? I said, I don't know, but I don't want to attend any interview. And at that time, there were no pharmacy companies that were coming in actually to give any kind of opportunities. Usually it used to be IT companies and fast FMCG companies like HLL and consultancy companies. These were the major companies who used to come to the campus for mass recruitment. So they used to bring a lorry, pack up people and leave. So everybody will get a job. So I decided not to take that road, and I decided that I'll go back to Chennai and let me figure out what I want to do. And but incidentally, I think BITS had a fantastic tradition of actually allowing us to do industrial training for about 6 months or 12 months. So if you're a single degree student, you can do 6 months. If you are a dual degree student, you can do 1 year of internship. So I chose SPIC Science Foundation, which was one of the premier most research institute in Chennai. They were doing fabulous work on a lot of agricultural biotechnology. And so my intent for choosing that was I wanted to be at home, obviously. And also, of course, I didn't know much about that institute other than the fact that it was one of the most sought after institute where people applied, and usually they had good things to say about the institute. And I think really my thought process behind a research career probably started shaping up around during this particular time period I spent in Chennai in SPIC Science Foundation. Until then, I was a pretty mediocre student, and I hardly attended college because in BITS we had this amazing thing. You don't have to, attendance is not compulsory. And so you could set up your own timetable. If you want, you can attend. If you don't want, you do not have to attend. So I tried attending classes, didn't do well. So I thought I will not attend classes, and I thought let me figure out whether I can do well. That also did not work out. And I took some of my seniors advisors very, very seriously. They said marks doesn't matter. It's only personality development which is very important. Then at the end of 5.5 years, I realized that I neither had the personality nor the marks. So I had to essentially rebuild everything. And this practice school or the industrial training concept which they had was one of the ways by which I could actually improve my GPA. Okay, because you get evaluated for it. But I really had a fabulous time. And I think Naren always talks about an inflection point. And it normally, you realize it after it has happened. And I think that was probably my first inflection point in my research career. And that is where I met, I consider him as one of my first mentors. His name is Dr. Narayanan, KK Narayanan. He was actually the director of the plant molecular biology laboratory for that unit. And he actually, the month I joined him, he was actually moving to Australia to pursue some I think he was on a Fulbright scholarship or some other scholarship where he got an opportunity to work in Australia for about 1.5 years to 2. But he refused to take me, actually, saying that I will not be available, and it will be unfair for you that you'll be left alone. So it's better that you find somebody else within the team with whom you can work. But one of my really good friend who's now settled in the US, her name is Kavita, she advised me that if you are working in that center, no matter what, try to work with him. Otherwise, there's no point in working in that center. So I insisted, and he eventually relented. And incidentally, his PhD student was there in the lab, and he said, "Why don't you kind of work with him? And I will be visiting on a quarterly basis, and we will see how this arrangement works. And in case things move on, we will see how to manage your workload." And so prior to moving to Australia, what he did is he had given me some basic molecular biology task. So, and that was one of the most well-equipped lab in Chennai at that time. So they had almost all the facilities that was available for any student, okay, from a college to get exposed to different kind of tools, analytical tools, molecular biology tools. And so I was very fascinated by all of this. So my first, I think, 3 months was just getting to learn all the techniques. And during that time, I would say, I think I never got any of those techniques correct in the first. So it took me a lot of time to actually learn to work in a laboratory setup. And many a times I used to get very frustrated because the results will not come. And I used to get very irritated. And so during one of these occasions, I told him that, I told him, is there anything else I can do? Because I'm not able to do what you gave me. So then he said, why don't you, he said, let's do this. So he used to have his office in the terrace where he had his own laboratory reagents. So he said, why don't you come and work with me? Let's do this over a weekend on a Saturday and Sunday. So here was a director of the institute who said, I'll just spend some time with you on a Saturday and Sunday. And what did he do? Nothing. He set up a concoction of enzyme reaction with restriction enzymes to cut the DNA band and run it in a gel to show me this is what you do. And that is what I was not able to get for the previous 2.5 months. And and then during the course of those 2 days, that weekend that he spent with me, he gave me a lot of small nuances in terms of reagent preparation, reagent handling, how do you handle the tips. And he was very finicky. He will not give us, he will lock his pipettes. His office room will be open, but his pipettes will be locked in the drawer. You cannot take that pipette out. Okay. Similarly, his tips. Okay. So I asked him, so you leave the office open, but why are you locking all of this? He said, that is a trick. It's very, very important that you need to take care of your pipette and tips, okay, to get the correct outcome. Okay. And then, so like this, he taught me a lot of nuances. And voila, after that weekend, whatever I touched in that lab turned gold. Okay. Whatever I couldn't cut with the restriction enzyme, it started cutting by itself. I was able to do a lot of cloning, subcloning. And subsequently, I did this, I was working on rice mitochondrial genome. So that was my first project that I handled. Where we were trying to prove a hypothesis that that mitochondria is actually a prokaryotic organism evolutionarily, which got actually engulfed by an eukaryotic system, and they learned to coexist. And so we were trying to prove this hypothesis by studying the rice mitochondrial genome. And I think I spent about a year, and he was mostly away. And actually, I started working very independently. I used to correspond with him when he was in Australia at that time, and I used to work with him. And whenever he used to come to town, he used to spend some time and talking about my project. And that is how I think I gained a lot of confidence and independence in terms of handling scientific problems. Then I shifted to sunny Buffalo for a program in pharmaceutical sciences. Actually, I joined the medicinal chemistry program first, but the medicinal chemistry program, eventually they were moving out of the School of Pharmacy into and merging with the School of Chemistry. So I decided to stay back in the pharmacy school, and then eventually I pursued the pharmaceutical sciences program for about 5 years. 5.5 years I was there. And I graduated in 2005. I think this was essentially my graduate school, my college and graduate school life. And during this phase, of course, I'm very passionate about music also. I started learning mridangam when I was at the age of 4, and I used to be very active in the music circuit in Chennai, which was the mecca of Carnatic music. So I got an opportunity to perform in many of the concerts, and probably I was more interested in that than my studies. Until my father pulled me up one day and said that, look here, I cannot support you. So you decide what you want to do. Okay, you cannot keep playing in all these sabas and these concert halls and think that you can make money. It will not happen. And we are not from a music family. So you need godfathers and godmothers who can push your career. So what do you want to do? Do you want to just keep playing mridangam all your life, or do you want to really study? So I said, okay, fine. I said I'll try to study and I'll try to be a professional, if not a music professional. But that passion never left me, actually. And in my view, I think until I finished my graduate school, until I came back to India, I think that was the biggest sanity factor in my life. I think in BITS I was very active in the Carnatic music group. We used to organize a lot of concerts. Even in Buffalo, New York, there used to be this Cleveland festival, the Cleveland Tyagaraja Aradhana, or the Toronto Music Festival. So people used to stop via Buffalo before going to Toronto. And so they used to have these chamber music concerts with all the musicians who would stop by, and whoever doesn't have an accompanist, they will ask me to come and play. So at least I was able to kind of keep me engaged. And I used to play in the temple because they used to give a lot of good food at the end of the concert. So that was also very important. So I don't have to cook and I can go there. So it kind of kept me alive. The music kept me really, really alive, and it catered to the creative needs of my brain. I think that was very important. So then I did my PhD in factory. Okay, I think I was the first student in my advisor's lab. So once again, when I spoke to my advisor, his name is Balasubramanian, he's very well known now. At that time, he was a research assistant professor. So he was not even in a tenure track. So in fact, both of us got the admission on the same day. He became an appointment. He got an assistant professor appointment, and I got my admission on the same day. And I said, I will work with him. So he was shocked. He said, why? I said, I don't know. I just want to work with you. I like your work, at least. It sounds very interesting. He said, there are other very established faculty in the department. Why don't you work with them? Because I'm just starting my career. And he was afraid that if his career doesn't take off, my career will also. So I said, it's okay. I think we have 6 years. So we must be able to do it. Something about doing things. We can do it together. So I was one of the first students in that batch where I directly joined the lab without even a lab rotation, and I started my research career. So being the first student in the lab, I think there were quite a lot of challenges. There was no precedent data. There was no precedent equipments, laboratory that was available. So they gave him a seed money. They gave him, I would say, the university had given him some seed money, and we shifted about 3-4 rooms, actually, which were makeshift labs for the first 2 years, until he got a permanent position, as well as and he had his own NIH RON grant, as they call it. And UB was very traditional in the sense that faculties were evaluated based on their NIH RON grants. They have to get it for a tenure track. You cannot have an industrial grant. Okay, you can have millions of dollars via industrial grant, but that was not considered for a faculty tenure. So obviously he was extremely, I would say, nervous about ensuring that he gets his NIH RON grant. And the first 2 years was more about doing coursework and setting up this lab and starting to work on Factor 8 and generating some data which will feed into this grant. And in that process, we tried to publish, and I think we got about 15 of our first approaches rejected. About 15 papers we got rejected. Same paper, different theme, multiple drafts, and nothing went through. Okay, but we still managed to get the grant. NIH R1 grant, we were able to get it to top up the seed money that you received, and we were able to get new equipments and everything. And I think eventually we got a breakthrough sometime in if I'm right, about 2 years into the program. We started collaborating with Dr. Russ Medoff in University of Kansas, who was a very well-known name in protein structure. He was actually a big vaccine formulator for Merck Biologics for a very, very long time. I think, Naren, you might know about Russ Medoff. And he was a big faculty in Kansas. And so our lab started collaboration. And I think through this collaboration, I think we had a breakthrough in terms of getting a couple of publications out. And one thing good about my advisor was that he never micromanaged. So he told me that, so he was a 9-to-5 person. So he will come at 9 o'clock, go in between for lunch, come back and leave at 5:30. So the only thing he used to say was, it is your life. You do whatever you want to. I cannot manage you. I am not your parent to manage you. You manage your time. You manage your work. Okay. At the end of 5 years, you must have a body of data that you should be proud of and you can defend and write something. So and he said, this is what my advisor told me in IASC. So he was a biophysicist by training. And at that and I think he was one of the first few people, at least in UB, who wanted to implement biophysical techniques for protein product development. And incidentally, he was the only person who did not have a PKPD background, although our department core was PK and PD. Okay. And one of the expectation from the department was that, no, you can do anything, but to graduate, you will need to incorporate PKPD concepts into your thesis. You should have exposure to in vivo animal studies, and you should be able to integrate it into a submittable thesis to the graduate school. So that was one of the mandate. It was an unwritten rule. So our journey for Factorate was very interesting. So it started off as a collaboration with Baxter, and then eventually something did not work out from an IP perspective, and we decided to move on. And we started working on liposomal delivery of Factor VIII, trying to reduce its immunogenicity. So it was a fantastic time setting up the entire infrastructure for Factor VIII and building on some of these protein immunogenicity concepts. In fact, we were one of the first labs to report that all protein aggregates need not cause higher immunogenicity. In fact, around that time, I think Amy Rosenberg in FDA, and I think Naren has worked very closely with her. So there used to be this overzealous discussions on how aggregate can basically totally screw everybody up, and it is essentially a poison waiting to basically end your life. And here we were looking at some data where Factor VIII aggregate, which was we had thermally aggregated the protein, we injected into the hemophilic mouse model, and it did not show immune response. It was a dampened immune response. And so we repeated that study multiple times, okay, because it was against all the publications that were available. Then, but we didn't know whether to publish it, because what do? We have to give some mechanistic basis of why we are seeing a reduced immune response, right, for this particular product, because it was growing. It was against the the then known literature in the scientific world. So during the course of our laboratory discussions, we said that is there anything in the protein whose antigens are essentially buried, because of which it is not engaging the immune system very effectively? So Factor VIII at that time was known to have certain highly immunodominant epitopes, which was capable of eliciting a lot of immune response. So in the thermally aggregated protein, luckily we had commercially available monoclonal antibodies, ELISA kits were available where you could probe some of these immunodominant epitopes. And it was surprising that in the aggregated protein, all those epitopes were buried. So it was not visible to the immune system to actually evoke an immune response. Okay, it was a very surprising element for us. And we actually published this. And I think it was kind of well received, although we were very skeptical that our paper could get rejected. We still went ahead and published. And another important aspect of my thesis was I was doing liposomal factor VIII, and we had phosphatidylserine as one of the components in those liposomes, because we wanted to mimic the surface of the blood platelets in which factor VIII will bind and elicit the coagulation mechanism. And interestingly, of course, we wanted to mimic the interaction of factor VIII that happens in vivo, which is a blood coagulation protein. However, what we didn't know at that time was phosphatidylserine itself had certain properties which was kind of suppressing the immune response, which we were not aware of. And there were very, very old historical literature which was available where there were some reports that phosphatidylserine can have an immunosuppression or an immunodampening effect. So we started probing that. Once again, liposomes are actually used as adjuvants to improve immunogenicity. Okay, if you look at all the vaccines today, the COVID vaccines that we have, they all have nanolipid. They are all called NLP, nanolipid particles. Okay, once again, one of the surprising findings that came out was our liposomal factor VIII had a reduced immune response even when given subcutaneously. So that was another that was another surprising finding that came out of that thesis. And once again, people refused to believe that. And so incidentally, what had happened is we in order to probe the mechanistic part, we actually took the liposomes, just the empty liposomes, okay, just the lipid particle, administered it, co-administered factor VIII, okay, and we saw that it was still able to reduce the immune response. So there was something in that liposome lipid composition which was dampening the immune response, okay, but we couldn't understand, or we couldn't, I would say, scientifically provide a basis for why this reduced immune response is happening. And I was nearing the end of my stay. Okay, so I said I wanted to graduate. So he said, okay, write up the thesis. And he said, this particular finding where you are co-administering the protein and the empty lipid without combining it, and it is still reducing the immune response, you can't put it in the main body of the thesis. You have to put it only as a kind of annexure, because this is a questionable data. I said, if it is a questionable data, why are you allowing me to put it in the thesis? Let's not do it. No. You've done that's a lot of work. You still need to put it in annexure. So I had this argument with him. Then he actually threatened me, saying that if you put it in the main body of thesis, I'm not going to sign off the thesis. So what happened is, of course, all this in jest. So we sent it to, so we had an external reviewer who was also very well-known liposome. He was very well-known in the world of liposomes. He was from University of Wisconsin-Madison, Dr. Timothy Heath. So he was very well-known in this area. So he saw this annexure, and he sent back his comment saying that until the data and annexure comes to the main body of thesis, I'm not going to sign off on the thesis. So it became a trade-off between my advisor and the external advisor. So I told my advisor, look here, I have to go back. So there is no way you can keep having these arguments. Let me put it, I will not publish this anywhere. We will not publish this until we know the mechanistic basis of all of this, which anyway, you ask the future students to do whatever they want to, I have to leave. So we agreed to disagree and move that annexure part of the thesis into the main body. Okay. And that was also the time something in me flipped, wherein I said, I just want to go back to India. Okay, no reason. Okay, I had interviewed in a couple of places. I had offers. I was very keen to get into Genentech, but Genentech did not even call me for a single interview, leave alone face-to-face. I didn't even get a single phone call. So I was very disappointed. And I got offers from Bayer. And on the day I was leaving, I said that I got a call from Sherimplo, which is now part of Merck, saying that would you be interested? I said I'm actually on my way back to India. And so when I told my advisor that I was going back to India, he was shell shocked. He didn't talk to me for 2 days. So he asked me whether there was any family problem that I had, whether I had any issues, whether he can help me. He was very nice. He wanted to he said, well, you can continue as a postdoc for some time if that is what you want and all that. But at that time, I was keen to get out of the lab. So I didn't even want to stay back. And then by the time I had finished interviewing in Biocon, and I just knew Biocon existed. I had no clue as to what they were actually doing. So I joined Biocon without visiting Biocon. I just packed my bag and came. And when I came in, there was no, once again, there was no protein formulation lab or a product development lab. Biocon was just getting into that domain. And they wanted people to come and establish labs. And that is how, actually, I would say quite a lot of my colleagues who are currently in Biocon actually joined during the time frame of 2005 to 2006. Very entrepreneurial nature in the company. You can do, I think we were allowed to do a lot of things wherein you can set up your own team, set up the lab, set up infrastructure, learn as you go along, learn as much as you can. Okay, talk to people, bring in all those talent and knowledge back. And I would say that I think that was also one of the, I would say, a second inflection point. So my first inflection point was my initial exposure to research in Spic Science Foundation. Then my decision to move back to decision to join Balu. Okay, I consider that as an inflection point because of the fact that being the first student in the lab, it was not easy. And you had to do literally everything, build the lab from scratch. Okay, there was nothing to look up to or build the data on. So it had to be your own data on which you need to stand. The third was essentially moving back to India. Okay, taking a big leap of faith without even seeing the organization. I don't think that was really please believe it or not. I don't think I will ever do it if I were to go back in time, but I did it. And I think probably and staying put in Biocon. I think there was something magical about Biocon in the sense there was enormous amount of vibrant, and animated discussions we used to have in terms of what we will need to do and how do we bring India on map. Because I don't think anybody believed that an Indian company can develop a complex biologic product. I think that skepticism was always there. And I think kudos to Kiran, our chairman, who invested in all of us, and our first president of R&D, Sri Kumar, who brought in all these talent and essentially nurtured us, essentially. No matter what skepticism people outside had, they had enormous belief that we must be able to do something out of India being in India. And I think that was one of the biggest drivers for us, I think for all of us. And I think after that, in my 17 years stay in Biocon, I have had, I think this is the fifth R&D head that I'm experiencing. Naren was the fourth R&D head. No, Naren was fourth. Yes, actually today is Anuj is the sixth. Sorry. So yeah, so Naren was the fourth R&D head, and I'm sure Naren must have shared his own journey in Biocon. And I think one of the most important things that all of the R&D heads had done over the years was they never disturbed the ecosystem within the R&D. They allowed us to kind of be very vocal about what we do, what we feel, what we state. And I think that scientific spirit was always encouraged in the R&D ecosystem of Biocon. And we were the first to get we got the first biologic product approved from India in the biosimilar space in 2017, which was a landmark for all of us. Something that nobody believed an Indian company could do, to an extent that 9 auditors from FDA actually flew from the US, came to our campus just to see whether we actually exist. Okay, I'm not joking. They actually said this in the audit. They wanted to understand, okay, through their own eyes, that there is actually a company which can actually do all of this. Okay, so they spent about 15 days, 9 of them. Okay, and few of them also happen to spend a lot of time in R&D. They're looking at all of the data, what we do, how we manage things, the workflow. And they were thoroughly convinced and impressed. I think when they left, I think they were totally, I would say, stunned that an Indian company can actually do this. And we have actually fantastic scientists who can contribute to such complex product development. I think that was one of the, I would say, and that barrier was very difficult to cross because it was about, and it involved about 10 to 12 years of work. Okay. And several missteps, okay, going back to the drawing board, okay, redeveloping. And of course, we received a lot of help along the way, okay? It is not like you require an army of people to do all of these things. We got help from various people. Naren put us in touch with so many consultants and scientists and his peer group who was able to give a lot of insights on product development. We had our partners in Viatris, that was erstwhile Mylan, who had come in, and they were also learning along with us because they were a small molecule generic company, not in the biologic space. So they were also learning along with us. And of course, we were a unique company in the sense we had insulins, we had monoclonal antibodies. So we had a range of product lines, okay, with range of complexities and range of scale of operations, okay. In my view, I think I don't believe, I don't regret one day coming back to India. Okay, now I would say somebody can say you lucked out. Probably yes, luck was on my side because when I left back, one of my classmates asked me, "What will you do if you don't like that place?" So at that time, I didn't realize that you can't go back to the US because your visa is out and they will not sponsor you again. I didn't realize that. So so when I came back with the mindset that I must learn to like what I do. I must learn to adjust, okay, to the ecosystem, no matter how difficult it might be. Okay, go with the flow, okay, and create your own niche and path. Okay, so these were the 4-5 things that I had in my mind. How I will do it, what I will do, I had no clue. But I think here I am 17 years into the organization and I'm a fossil of Biocon now. To a large extent. And
So great. I think a fantastic story. And, I'm sure there's much more to tell, but maybe I'll hand it over to Charu to sort of coordinate the Q&A.
I have to say that was a wonderful and such a passionate insight into your life and views and your reasons behind several decisions. And you've given us so much to think about. And I think that there are several questions. So I would invite everybody just to raise your hand and Vikram will announce the lucky first questioner. Okay, well, I mean, I guess if it's okay, Vikram, should I ask a question?
Yes, please, Shah Rukh, go ahead.
Yeah. So, Karthik, I wanted to ask you about your research in mitochondria. So did you finally reach a verdict? Because there's still much confusion, like what did your story tell? Is mitochondria prokaryotic symbiote?
I think we were inching towards the fact that it was essentially a symbiotic relationship between the mitochondria and the eukaryotic system. But once again, I have not touched this topic after 1999. Okay. So which is I'm now 45. So 1999, and I celebrated my 25th year of visit to Pichplani also. So I have no clue how that area has actually progressed over the years. But yes, we were really close to actually proving that because we did map few genes which had migrated from mitochondria into the rice genome. Okay, so and I think so we were looking at basically certain migration of certain specific genes over the evolutionary period of mitochondria coexisting with essentially the eukaryotic system, and we had mapped a couple of important genes. But that is when I think that project also died with that. I think it didn't proceed. We didn't continue in that project. And I think eventually SPIC Science Foundation closed after a couple of years. The answer to that question is probably if we had continued, maybe we would have come to a logical conclusion. But I'm sure there are other groups which would probably have an answer to this. I'm sure, because it's 1999 and we are in 2022.
Yeah. Thank you.
Yeah. Okay, Virender, you can go ahead.
It's nice to hear from you. Like, I think I was there from 2008 to 10 in Biocon. I was working under Sudhir in quality control and mostly managing insulin in process team. So, and I could relate with like many of your like stories from Biocon and audits. So I was there like when Pfizer was like auditing for collaboration, but I think when I left, so that collaboration came to an end, and Biocon got a lot of money at that time. So I just wanted to know, like, is any plan when it comes to recruitment for people who are like doing postdoc or PhD from outside India? We always.
I think today, if you look at it, there are quite a lot of people. As I said, during that time period I joined, there were a lot of people who had done their PhD in India, but they had gone out to do their postdoc, and they all came back. And many of them joined around this time frame of 2005 to 2008, and many of them are still there along with us. Along with me, they continue to be part of the organization. And one of the reason I think we have, I would say one of the reasons why, at least I cannot speak for others, but what has continued to stay in this organization is also that I think, first of all, I think amongst the Indian companies, I think the scientific temper in the organization is very, very high, at least in R&D. Okay, I can speak for that. The facilities that we have, okay, the level and depth to which we can challenge ourselves. Okay, Naren would have witnessed literally fist fights happening between all of us during the time when we were getting our first approval. Literally. Okay. So Naren used to call me as a problem child when he first saw me. And I used to create a lot of ruckus. Okay. And then he tried to change me. Then he said that, no, I will change for Karthik. So that's what he did. So literally, and that is because of the fact that we used to be so passionate about what we used to bring to the table, and literally, rip apart, okay, our work, okay, because at the end of the day, the goal was, okay, to stand you can fight amongst yourself, but you need to be able to, defend in front of the outside world. And I think that scientific temper still continues, maybe not to the same extent because of a lot of changes and dynamics. And also the time frame that Naren was there, we have not gotten any product approval. Okay, now we are more a commercial organization. Now product approval has become a second nature for us. Okay, and now we are in a very different trajectory where commercial pressure is driving the organization in many ways. And when you have that inflection moving from science and development, not that science and development is not important, but I think the commercial pressure and the decisions, okay, that happens back on the science and development is to a large extent dictated by how your partners and the investors view about you as an organization. So I think we are in a kind of a very different trajectory. So, but yes, we are open to, we still scout for people from outside, okay, with PhD experience, with postdoctoral training and experience. Yeah, but one thing is very clear, we are not good paymasters, okay? So that is going to be a big challenge, okay? So if you're looking for basically, if you compare a DRL versus a Bacteria laboratory versus say a Glenmark, or for that matter, even an Intas versus Biocon, we are not, in terms of the pay, we are not on par with them. But there's also a reason for that. The reason is all of those companies, they are basically very well known in the small molecule world, okay, small molecule generics, okay, and biologics R&D is an extension, okay, investment of their core business. Whereas Kiran was very different. She made biologics the core business. And even before anyone in India, okay, could commit so much amount of capital intensive money, okay, to basically make progress in biologic space. Okay, she actually invested so much money in the biologic space. Okay, that is not easy. Okay, even when you don't have a massive revenue like the Reddy's and the Sun Pharma and your Glenmarks, okay, she was very committed to investing the money in the biologic space. So, so I would say biologics has always been the core of our existence. Okay, and we started off as a brewery company, right? We are a fermentation company. Okay, we used to manufacture enzymes for brewery industry, leather industry. Okay, and then fermentation runs in the blood of Biocon, in the DNA of Biocon. That is the platform technology that we have leveraged when we have made from one transition to another, from enzymes to statins to essentially biologics, from insulins to monoclonal antibodies. So that has been the core leverage in terms of technology. And so we were very different even in terms of approaching the biologics, what you call product development and biology and commitment to biologics. And I think that is probably one of the reasons why people like me continue to stay there, because of the science, the complexity, as well as nobody does what we do. Nobody does the operations at the scale at which we do, especially in the insulin space anywhere in India, or for that matter in Asia. We run fermenters at 50 KL. 50,000 liters. Okay, whereas in the monoclonal antibody world, we are talking about you will have multiple 6,000 liter tanks, whereas we have a single 50 KL tank here. So you can imagine the volume at which we operate, the scale at which we operate. So for a technologist, okay, for a process engineer, for a scientist, imagine you get to work on almost from the most complicated molecule, okay, to the most complicated scale of operations. Where else would you get this? So I think that is what has driven me to continue to stay put, in spite of all the other things that happens in your life within the ecosystem. Because after some time they will take you for granted. Oh, this guy is not going to go anywhere. He'll be around. So, so that kind of mentality also sets in. But overall, I think the organization has been very, very nice, and I've been very happy to be part of the organization.
Okay, Sunena, and then Shruti, you can go.
Hi, Dr. Ramani. Thank you for your insightful discussion. My question here is, what are your learnings from the mentors that you have had in your life? And what is your take on being an ideal mentor? Okay.
I think all of the mentors that I have interacted with, they have enormous passion. I think that has been one common thread in different ways. That passion comes out in different ways, but that has been one very key thing. Okay, even for example, I consider Naren as an important mentor. Okay, and I know how passionate he is about certain things. He's very passionate about his music. He's very passionate about people in general. Okay, and he wants people to be given that space to flourish. Okay, so they exhibit passion in different ways, in different dimensions. So that has been a very important thing. And I think if you are not, if you cannot be passionate about what you're doing, or I don't think you'll be able to inspire anybody, or you cannot basically be, you will not be able to rub off or essentially make people understand or believe in you. Okay, so I think that is very important for a mentor in my view. And I think another important thing is what I've learned from my mentor. I think passion. I think dedication to their craft. Okay, I think definitely, how do I put it, pushing themselves, okay, and asking the right questions, probing, okay, I think that is very important. Enormous conviction, okay, I think that is very important. I always tell people, you need to have conviction in what you're doing. Okay, if you don't have a conviction, and that conviction should be built on data. Okay, so I always say, as Narayan Murthy says, in God we trust, rest is data. So I think there's nothing truer than that statement, especially in our area. Okay, and if you don't, and we can, I think we have all witnessed the way COVID pandemic has been managed, okay, across the globe, and what lack of data can do in terms of policies and in terms of basically how do you manage, okay, the response to this kind of a pandemic. Correct. Conviction, and then pride in what you do. Okay, if you don't take pride in your work, nobody else will take pride. Okay, so you have to sell your work. Okay, that doesn't mean that I'm saying you need to basically take a trumpet and put a drum and keep talking about it. But I think it's about standing up with a lot of conviction and a lot of passion in terms of what you believe in, and essentially, what do I say? Um, Fighting for the right reasons. I think that is very critical. And also speaking your mind out. I think that is very important. Okay, and sometimes we are all constrained by our own circumstances in terms of not wanting to speak your mind out. Okay, at least I have not had that problem, and I think Naren can also vouch for that. I've been very vocal about my thoughts, typically across the spectrum, not to kind of take a stand, which is basically essentially which is counterintuitive or anything. But I think the important thing is if you don't speak up, okay, see, if you cannot fight for yourself, how will you fight for your team? Okay, that is very important. Okay, you are going to be leading a team. People are going to be looking up to you, and if you cannot fight for yourself, okay, how will you fight for the rest of the people who are looking up to you? So I think that is very important. Okay, so fighting the right battle, okay, speaking your mind out. And I always tell people, I always remember what my chairman of our Department of Pharmaceutical Sciences in Buffalo said. He said, when I left him, I left in 1999. So he said in his 30 years of existence as a chairman in the institute, he had never seen anybody leaving the US immediately after finishing that PhD. So he said normally graduate students come in, they say they want to go back and they never go back. So you are the first person, at least to my knowledge, where I've seen see you going back to your home country. So, and he said, just remember one, he said, you are not going to be in a popularity contest when you go back. Okay, so if you are right, you can always defend yourself. Don't try to basically please people. And I think that was I think that has really kind of shaped up my approach to things. I don't try to please people. If they get pleased, fantastic. It's a bonus. If they don't get pleased, okay, sorry about it, but too bad. That is not to say that I'll just randomly pick up a fight, okay, or just for the sake of it, provocate people, not like that. But I usually speak my mind out, and I think that is very, very important. Okay, and it has worked, at least in my favor. Over the years, I can say that.
Yeah,
so I think to answer your question, what do I look for in a mentor? Passion, pride, conviction, and and most importantly, I think all the people who have been my mentors, they've had this enormous humbleness to kind of show me the go out and seek what you want. Okay, enabling. I think being an enabler. I think I always consider Naren as a fantastic enabler. Okay, and he has been very selfless that way. Okay, for example, he will just randomly pick up and say, why don't you talk to this person? See whether you can basically interact with them, collaborate with them. So nothing expected out of it. Okay, so I think these are some of the qualities I believe should be there in a mentor.
Yeah. Vikram, if it's okay, if Sunaina and Anuradha can ask a question, and then I'd like to say a few words before Shruti does her thing. It's okay.
Sure. Yes. Please.
I think Anuradha can ask a question.
Anuradha can ask a question. Yeah.
Okay. So I have a quick question on liposome. I'm not a liposome expert, but I want to know what was that specific reason due to which others were getting immune suppression due to liposome and you were not. And usually, according to my understanding, liposome is now being used with the vaccines as a carrier, and also what is that specific reason or property of liposome where we need to work or modulate?
See, I think when you talk about liposomes, right, I think it's a particulate matter, correct? So when you put a particulate matter in the body, okay, in a subcutaneous region, for example, it is rich in essentially antigen presenting cells. You have dendritic cells, okay, which can actually, it can attract all of these antigen presenting cells. Okay, so that is why normally they will give most of these vaccines, they have this particulate nature, okay, so that it can attract the immune cells and present it to the immune system more effectively. And that is also one of the reasons why liposomes are known as kind of adjuvants to boost the immune response. Okay, so even the COVID vaccines from Moderna, from Pfizer, okay, they all have cationic liposomes, okay, encoding the mRNA. Okay, so one of the reasons is it presents itself, the antigen, to the immune system in a very, very effective way, so that the body can mount a good immune response. Now, in our case, we saw the reverse happen, okay, even via the subcutaneous route. And what we later found out was that phosphatidylserine, which is present on the surface of the platelet, that has an inherent immunosuppression properties, which kind of enabled us to kind of reduce the immune response against factor VIII. So we were working on a multi-pronged approach. One was to see whether we can kind of mask the immunodominant epitopes, which are capable of evoking an immune response. Then we also wanted to increase the, the half-life of factor VIII, so that you don't have to keep giving the protein, because continuously giving the protein can also keep kind of stunning the immune system to evoke an immune response. So we wanted to reduce the frequency of administration. So, but eventually I think it was kind of serendipitous that phosphatidyl, and then we went back and there was some indicative literature which just said that the phosphatidylserine head group has a capability to have an immunosuppression effect. And in fact, that paved the way for the next wave of work that the lab started working on eventually. So I think now the lab has done fabulously well over the years. A lot of students have really taken the lab and the programs to basically to the next level in terms of probing this mechanism. And now our lab actually has generated some of these in vivo dendritic cell models, which can be used to predict immune response. So yeah, when we started, obviously we were not aware of this. Now I can tell back things in sequence. But at that time, I was scared that I will not get my thesis published. So I was more interested in graduating. So honestly, the wisdom with which I'm talking about some of these events certainly did not exist at that time.
Okay.
Yeah.
Okay. Sorry,
a
very quick question. What about the apoptotic cells? Because they also express.
What about the?
Yes, apoptotic cells undergoing apoptosis, because they also express PS on their outer surface. So do they still bind to liposomes?
No, see, phosphatidylserine is one of the, it is a lipid component. Okay, so we artificially prepare these liposomes ex vivo, okay, and entrapped factor VIII. So essentially, or other intercalated factor VIII, because there were specific regions in the protein which was capable of binding to the liposome surface. It was a charged surface. Okay, so I think essentially and by nature, phosphatidylserine also gets cleared away very rapidly. Okay, that is also one of the reasons you have to pegylate the surface of the liposomes. Okay, because phosphatidylserine has a tendency to get cleared away by the body. Okay, so normally, and the primary organ which clears this is the liver, hepatocytes. Okay, so that is why I think when we started doing this work, also conceptually it seemed very, I would say, from a concept perspective, we thought that things will work forward. There are a lot of things in the literature which did not support that our concept would work. Okay, and I think it worked. The reason it worked is because of the uniqueness of factor VIII and the uniqueness of that interaction with those phosphatidylserine liposomes. Okay, that is very unique. Now, I don't think any other protein liposome interaction with this composition can work the way it worked for factor VIII.
Yeah. Well, thanks. So we can dive into all kinds of things with Karthik, but I just wanted to say a few points before Shruti takes over and does a rapid fire with you, Karthik. But one of the things that, as you were telling us these stories, I think one of the things that really apparent, that I think you will all see, that Karthik doesn't say it, but he has enormous amount of courage, and that's something that I really admired in him, despite everybody being against the idea that he was bringing forth. He believed in certain ideas, and like pig model, for example, Karthik. I mean, I think that was a fantastic idea, but we never ever could do that experiment. But sometimes
I have still not lost hope.
I know that. But I think sometimes in the lesson for me that I learned from Karthik is, believe in what you really want to do, and stand behind what you really want. And that's courage is something that, Karthik, I really, when I see you, I think of that word. So
thank you.
That's what I want.
Yeah, and now I'd like to invite Shruti to start the not copied from any talk show rapid fire round.
Thank you so much. Hi, Karthik, I'm Shruti. So I would like to ask some quick rapid fire questions. Okay, first, life in USA versus life in India. What do you like the most?
Life in USA versus life in, if you talk about quality of life, yes, US is way better than India, no doubt on that.
What do you think? Reservation is a boon or curse?
I think when used appropriately, it is a boon. But the way what I see in India, it's definitely a curse.
Mm-hmm. Okay, so if you got a superpower, what you would like to possess?
What I like to possess,
Yes.
I think some a wand which can kind of completely reformulate the public health policy in India.
What is the one thing that you are most afraid of?
Most afraid of?
Yes.
Most afraid of? Well, sorry, it's a rapid fire, but I'm still afraid of. Honestly, I've never been afraid of anything. Actually, I don't. Sorry, I cannot answer that question because I've really not been afraid of anything. Yeah, maybe. Yeah, you can say ghost. But other than that, I don't think I've been afraid of doing anything.
Okay, great.
It validates my statement on courage.
Okay, so you liked spending time more in academia or industry? Because I believe that you spent equally in both.
No, actually, last 17 years has been in the industry. I don't think I'm cut out for academia. Definitely not. I think I need. I would say, for example, I think I'm more applied in my work. And I believe academia is very important. I think they do ask a lot of fundamental questions, which is very, very important. But I don't think I have the patience to run a run a lab, write a grant. I think that's a lot of work, and then go through this publish or perish culture which exists in academia. I don't think I am cut out for that. So my I mentally, I don't think I'm geared up for that. So I'm more biased towards the industry. Not that it is fantastic, but I'm willing to live with the industry compared to academia, because that's a lot of hard work, definitely.
Okay, what advice would you like to give to your 25-year-old self?
My 25-year-old self, what I would do? I think I don't know. Maybe I should have pursued Mridangam as a profession. I could have probably done, probably a musician. Although I think, if not a musician, I think I'm passionate about law. Okay, my father was a lawyer. Okay, we used to argue to death on any topic. Never used to agree on anything. And I think that was also one of the reasons why I'm very argumentative in nature. I think all of you would have read Amartya Sen's book, right? The Argumentative Indian. I think it goes very well with me. So I argue on everything. Sometimes knowing even that I'm wrong, just to kind of keep the topics open. So yeah, I would have loved to be a lawyer actually. And I always have a grudge against my late father for not advising me properly, although he was a lawyer, that I could have pursued a lawyer's profession.
Okay. If a movie is based on you, what would be the theme of that?
Movie based on the theme.
Yes.
I think I can say somewhere it should be a culmination of art and science together.
Okay.
Because I think
because
the percussion instrument that I learned, I think is very what do you call? There's a lot of intricate mathematics involved, and I think I don't know. It's very difficult. I can never be a good teacher of that instrument, but the way I process the information and apply it when I accompany an artist or what I need to do, I think that is kind of I think it is built on my scientific career also. So I think somewhere a theme which can integrate both of this will be probably a theme I can consider.
So if you want to become an expert on something, what that would be? What are you expert in right now?
Currently, I think I've moved away from expertise thanks to Naren. He only, before he left, he changed the organogram and left. So I was a drug product expert wanting to explore that drug product area, drug product development more. And before Naren left, he organized the R&D in a way which allowed us to get a breadth of exposure. Not that I regret it. I think it has been fantastic even then. But yeah, I think probably in a I would say drug product development is what I would like to consider my expertise amongst all the things that I'm aware of, relatively speaking.
Okay. And if you could teleport somewhere, what that place would be?
Teleport. Teleport. I think I want to visit this one, Finland.
Okay. And final question, what would be your lifelong dream? Lifelong dream. I mean that.
Lifelong dream.
Yes.
Lifelong dream. I don't know. I think, as I said, I've been talking to Naren also. I've broached about this topic. I really want to move away from industry into public health. I don't know how to make that transition. And so I think hopefully I will be in a position to influence some of the public health policies of India in a more meaningful and a measured way. I don't know how to do the transition. But yeah, I think putting myself in a position where I can impact the public health policies in a meaningful way, I think that would be something that I'm aspiring for at some point of time.
It was really great asking you questions. Yeah, thank you so much.
Thank you so much, Karthik, for giving us the opportunity to speak with you. And it's these rare Tuesdays, that we get to meet all of these big people like you. And I think
certainly
all of
not
us.
Big. I'm sure Naren had organized a set of people who are far more accomplished than me. So.
Now, see that's your humble nature.
And this is my daughter.
Oh.
Hi.
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 Her name is Mrinmay. Say hi.
So good.
Okay. She wanted to show her painting. Okay, it's beautiful.
Beautiful.
Yeah, and we've had so many questions, and we have so many more questions. Is it okay if people write to you on LinkedIn or on your email? Which one?
Absolutely. My email, Naren has it. Both my Gmail.
We have your email, so we'll just send it across to everyone who attended the session.
Absolutely. Anytime. Please feel free to reach out anytime.
Yeah, thank you so much. And
Thank you.
this was a great opportunity.
And thanks for patiently listening to all of this.
Great. Thanks, Karthik. I'll catch up with you
Bye-bye.
soon.
Thank you.
Bye-bye.
Thanks. Thank you.
A network should last a lifetime. Let us help you create lasting professional relationships with our world-class mentors through the Biopatrika Industry Mentorship Program. A strategic guidance program unlike no other, full of expert interviews, industry internship opportunities, CV writing, inflection point analysis, life maps, and of course, the gateway to your dream career. For a limited time only, all our services are freely available for you as we truly want you to succeed.